when we try to separate the endemic sites and look at the relative proportion of antibody against a specific antigen, and compared between the 5 endemic sites, there were significant differences. And our p-value is 0.005 because we wanted to be sure that this was not something that was due to chance. So what we saw was that if you look at the antibody of the ODs or the relative proportion of antibodies against EBA175, there were significant differences in a high endemic area like Kintampo and Navongo versus a low endemic area like Mali. And we saw something similar in EBA181, where high endemic areas had higher relative proportions of EBA181 versus lower endemic areas, and something similar in RH2, where Navongo and Afar were distinctively different. Surprisingly, or more interestingly, in terms of RH4, there were higher levels in the lower endemic areas, in the lower endemic areas versus the high endemic areas. And this was very, very significant. So the questions that we want to ask are, at this point, what do these differences mean for the development or for the search for a vaccine candidate which can produce strain transcending neutralizing antibodies, despite all this variation in the levels of immunity or the levels of antibodies against these specific proteins. So we also looked at the breadth of antibody responses, and as almost as expected, in Accra, which is a low endemic area, there was the majority of children there had little or no antibody response. Versus Kintampo and Navongo, where the majority of children there had about 2 or 3 antibody responses, and it was more sporadic in Hohoi and Mali. Then we wanted to find out, okay, how do these OD levels correlate with parastemia? And we found that there were significant negative correlations. Almost all of them lit up quite significantly, with the exception of 3 of them. So there was a significant correlation between the antibody ODs and the parastemia. So for future work, what we intend to add on to this work in order to further understand will be the ligand gene expression work, which will tell us the expression profiles and how they relate to these antibody levels, as well as invasion assays and growth inhibition assays. I would like to thank Dr. Wandari and the members of the Conway Lab at LSHTM and the Drakeley Lab at LSHTM, some of whom are here, and Gavin Wright for giving me the proteins, Francis for Synthesizer for expressing them for me, and James Beeson for allowing Kevin to give me some of his proteins as well. Thank you.
Yeah. So this speaker really beat the time by 2 minutes. That's good. So I have a quick question for you. Your statistical analysis, how many repeats were that? I didn't see that.
For which one?
The people.
For the graph.
Yeah.
So it's the relative proportion. So what we did was that we took a single individual's total antibody response, right? And then we made it a proportion of a single antigen. So your response to a single antigen as a proportion of your total antibody response, so that we could compare the levels across the sites, so that there'll be no bias in terms of endemicity.
Okay.
Henry, this is an unfair question, but it relates also to the last speaker, Emmanuel, although a different type of thing. I'm Keith Gull. I work on trypanosomes. I'm not an immunologist, but it always strikes me odd when people put up slides where yours are about proportion of the antibodies, and it's about a difference of about, it's 0.1 to 0.2. Emmanuel's is sort of like the levels of antibody, and it's sort of 5 to 10. So that sort of says to me, what's the biological sort of likelihood, possibility that that's significant? Might be statistically significant, but in biology, in my body, you know, if I have 2 antibody molecules rather than 1 antibody molecule, or a mixture of these things, is it going to make much difference? So, I mean, it's unfair because I ask this question all the time in the Dunn School of Pathology in Oxford, and I don't get much of an answer. But it's statistically significant, I agree, and your proportions are different. But 1 to 2, you know.
To be honest, I can't give you an answer.
Well, I do this mischievously because it would be good at some point, maybe if the immunologist could teach me a lesson in whether or not these are biologically significant differences, because I really doubt it.
Okay, someone is going to be
Not now, over a beer.
over here. That's all right. Okay.
My name is Julius Sabal from the London School of Hygiene and Tropical Medicine. Can you just comment on the results on RH5 and you didn't get any negative correlation between antibody levels and RCT, knowing that RH5 is probably the best candidate we have right now in terms of blood stage vaccines?
Yes, I don't know why I didn't get any correlation, but the sample size is about 600. So I think it's pretty robust. I will recheck the figures, but I can't tell you why there's no correlation.
We have room for one question. Yes.
Okay.
So in modeling this, one thing to think about is the cumulative effect of the different responses. What we tend to do is look at them individually, right? But yet we know that it's probably the polyclonal nature of multiple strains in which you need protection. So mathematically, what you should do is put that into a model in which you have something like area under the curve, and look at the summative effect of those different things, and the results may emerge a little differently.
Thank you.
Okay, so the plenary speaker, I think someone is going to give a little talk.
Okay, before we get to the plenary speaker, my co-commander has told me that if you check your program, immediately after the plenary speaker is supposed to be a coffee break, and then 2 more fellow talks. We're going to change the order of that. So it'll be plenary speaker, 2 fellow talks, then we go to the coffee break. Okay?
The speaker, Professor Solomon Oforiafa, is a product of Adisada College, which is not quite a distance from where I was, a much more superior college. And here at Mexico. Yeah, Professor Oforiafa is Associate Professor of Medicine and Human Genetics. He is also the director of a center in the same university for translational and international that has to do with sickle cell. He holds several NIH grants, training grants, and he is quite accomplished in his area of research. He has over 100 publications, chapters in books, and abstract presentation at conferences. And until recently, he was the president of the Ghana Biomedical Convention, and he was also a founding member of that convention association. So his presentation will be on engaging Africa fully in the global genomic revolution. I read the brief from Being a Continent. So, you're welcome, and give him a round of applause.
So whilst he's doing that, can you hear me from the back, or do I need to use this? I need to use this.
Oh, you have a loud voice. Go ahead.
So let me join the previous speakers in acknowledging the fantastic job that I didn't do that. the fantastic job that Gordon Awandori is doing, and our chair referenced my role in the Ghana Biomedical Convention, which we established 8 years ago. And I got to know Gordon because I think about 3 years ago we invited Gordon to be our keynote speaker. And in perusing his CV, I figured this is certainly somebody that I needed to know a little bit better. And so we did. But unlike Douglas, I haven't had the opportunity to have beer with Gordon about my presentation. So I still think it's 1 hour presentation. I haven't cut it down. I'm going to give you, okay, let's start before we have any other additional. So I'm going to try and give a talk that is really, it's not a scientific talk, but there is a little bit of science at the end. It's really going to give you a flavor, hopefully, of molecular medicine, where it actually started, the little bit of genomic revolution that has taken place, and how Africa fits into that. And I'm going to use sickle cell disease really as the backdrop, given that it was the first disease to be defined at a molecular level, and still carries the tag as the first molecular disorder. Let me acknowledge where I'm from, University of Pittsburgh. This is the University of Pittsburgh Medical Center. I'm in the Vascular Medicine Institute, and within the institute is where the Center for Translational and International Hematology that I direct is located. So by way of introduction, I'm going to talk about really sickle cell disease to give you a flavor of the basis of the disease, how we take advantage of that to diagnose it. And then I'm going to spend a little bit of time talking to you about the molecular pathogenesis of the disease. And then really identify the number of clinical complications of sickle cell disease for which we still don't know how to treat, and recognizing that it's a critical barrier in managing these patients. Then I will go through a bunch of slides, some of which I borrowed from the Human Genome Research Institute in the United States, to highlight what people tend to refer to as the revolution in genomics research. And then I want to spend the last part of the talk highlighting a rare opportunity that we have across Africa to really engage in the global genomics research, partly because of the type of diseases we have here, and more importantly, because of the large cohorts that we have here that geneticists are beginning to appreciate is a rich resource for discovery. So, in 1949, Linus Pauling published this paper in Nature. For students of basic biochemistry, this is just a paper on electrophoresis. And what Linus and his colleagues, Atino and the rest, Atino here, were able to demonstrate is that if you take red cells, well, forget red cells, if you take blood and just lyse it with water and run it on the gel in face of the paper, you get an electrophoretic pattern that is different from somebody who is quote-unquote normal. Normal here basically means they don't have a hemoglobinopathy, and somebody who has sickle cell anemia. And if you mix the two blood samples together, you get this sort of pattern. And if you get somebody who is potentially a sickle cell trait, you get this pattern. Now, this in 1949 was sufficient to win a Nobel Prize. I wouldn't try that in this century. You're not going to get a Nobel Prize by just running a electrophoresis. But this was pivotal because sickle cell disease had been around for years. This was 49. The first description of sickle cell disease in the Western world was 1910. And yet, it was unclear as to exactly how the disease came about. Some people thought it was the red cell membrane. There were so many ideas as to how the sickle cell disease basically comes about. Linus Pauling and colleagues clearly showed that, at least in this experiment, the hemoglobins were different. We now know, studying, and this is part of the work Max Perutz, who is one of the top crystallographers, and he actually figured out the structure of hemoglobin. and through a lot of X-ray crystallography, we know the molecular basis of sickle cell disease. Essentially what happens is that typically, so this is a protein fold, typically this position of the beta globin chain, or beta globin polypeptide, is occupied by glutamic acid, which is big and has charge. And when hemoglobin gives up oxygen, there is a conformational change between the molecule that brings in close proximity this region of the molecule to this hydrophobic pocket. So when this is glutamic acid, there really is no interaction between these two areas. But when glutamic acid is changed down in sickle cell hemoglobin to valine, which is smaller and sticky, you get an interaction here which begins the polymerization process. So if you learn nothing at all from my talk, understand that the basic molecular basis of sickle cell disease is that glutamic acid is small and sticky, and when oxygen is released from hemoglobin, it sticks to a portion of the hemoglobin and starts the polymerization process. You get a fiber that is actually stronger than collagen fiber, and this leads ultimately to red cells that get injured from within the cell, and these cells then adopt the so-called sickle shape. This really is the molecular basis of sickle cell disease. Now, if you look around the world these days, sickle cell and the sickle mutation, it is concentrated in Africa, but there are parts of the world where there is some sickle gene. I did my PhD in the UK. Where is it? Almost, that's exited the map. It is there. There is some sickle cell in England. But you can see that the majority is centered here. These are estimates of sickle cell anemia, homozygous sickle cell disease. It does not account for hemoglobin SC, which also causes sickle cell disease. You see that the majority of this really is in sub-Saharan Africa. And I'll come back to that as to why this, obviously, is an unfortunate situation, is an opportunity to basically engage the rest of the world in terms of genomics research. So if you do a simple electrophoresis, somebody who has essentially no hemoglobinopathy will have primarily hemoglobin A, and a little bit of, this is a minor adult hemoglobin called hemoglobin A2. And then there are four types of four scenarios where you get sickle cell disease. This is sickle beta plus thalassemia, where the individual makes about 35 to 30%, depending on the mutation. There are over 250 mutations that will cause this. And then they have sickle hemoglobin. This is a relatively mild type of sickle cell disease. It is still sickle cell disease. There are individuals who do not make any adult hemoglobin at all, typically due to deletion. And these individuals make primary hemoglobin S. They are as severe as the homozygous sickle cell disease. And in parts of West Africa, we have another variant of hemoglobin called hemoglobin C, which in combination with hemoglobin S gives you hemoglobin SC disease. So this is the disease. And so this slide, I'm going to try and summarize what's happened in the last 100 years. We do know about the karyotype. We know the chromosome that the gene, beta globin gene, sits where the single point mutation occurs. We know that. We know that it is an A to T single nucleotide substitution that leads to the substitution of glutamic acid now with valine. This is sufficient to cause the polymerization I talked about, sufficient to create this kind of black picture. There are two basic cellular abnormalities of a sickle erythrocyte. A sickle erythrocyte sticks very easily. Those who do malaria talk about malaria, rather red cells that are infected with parasite being sticky. Well, you haven't seen anything yet. A sickle erythrocyte sticks. It sticks to leukocytes, it sticks to endothelium, and that stickiness is what we believe causes vaso-occlusion during times of stress. And that vaso-occlusion essentially means that there is no blood flow downstream of this occlusive site, which means there will be hypoxia, and so on and so forth. So the first cellular abnormality is that the red cell, the sickle erythrocyte, sticks a lot. The second is that it actually lies within the intravascular space. A normal red cell has a lifespan of about 120 days. A sickle red cell probably around 20-25 days. It lies in the intravascular space, not extravascular hemolysis, which means it's releasing the contents of erythrocyte in the circulation when it's not really supposed to do that. We believe these two major cellular abnormalities lead to the myriad of clinical complications which affect all organ systems, from the head to to the toe. There are complications of sickle cell disease that you find. I focus primarily on the lung, and I'll show you some of the work we're doing on the complications called picture syndrome. I don't see that. Make sure you get this. So, being the first molecular disease, and given the advancement in molecular medicine, the question is, can we apply what we've learned from analyzing and studying DNA to address this? Because the only thing we've been able to do so far in sickle cell disease is we can diagnose the disease. There is no targeted therapy. We give infants, identify the newborns, we give them penicillin to make sure they don't get infection, but that's about it. Everything else is modifying therapy, whether it's transfusion, whether it's hydroxyurea, it's all modifying therapy. So the hope is that the information garnered from extensive analysis of the human genome might give us some insight into how we might be able to deal with this problem. So then comes the idea of genomic medicine, which you get people banding about. But it's really an emerging science. It's not a science that we are practicing. It is the idea that there will be somehow use of genomic information, particularly to an individual that you can use to diagnose them. And as I said, in sickle cell disease, we can diagnose. So that's not a problem. But in therapeutics, are we going to be able to use this in decision making? This is what the field of sickle cell disease really is looking for. So this then brings me the question, what is genomic revolution? What is it? I just want to start with a simple definition of what genomics is, which is very different from genetics. I'm not going to read that out. You can see it, reasonable size font. But it's important as we talk about this to at least know what DNA looks like. And I put a couple of these slides in just for students in the audience. DNA looks just like that. It doesn't matter whether it's from a dog, a plant, bacteria. DNA basically looks essentially the same. It allows us to pass on information from one person to the other. When did this actually start? When did the genomic revolution start? I think if you're a monk and you're studying how different peas give rise to different traits of a pea, that's pretty revolutionary. And so this is Gregor Mendel that I go back way back, and this slide I will acknowledge didn't come from me. This slide came from Francis Collins, who is currently the NIH director. This is the wrong slide, but I'm gonna go ahead. It's changed a little bit. I want to point out some of these are key things. I just want to jump straight to this here, the Human Genome Project. Okay, so these are the landmarks, and the slide I was actually supposed to show was for these red ones to then come on top. Because Francis Collins' landmarks in genetics and genomics, in my view, actually missed out the key ones. And the key ones were the description of sickle cell disease in 1910, Pauling describing the first molecular disease in humans, Maroto et al. In 1977 cloning the first cDNA from reticulocytes and sequencing and identifying the nucleotide substitution that caused sickle cell disease. And my favorite, in terms of landmarks, is the very first case of human identification ever by Sir Alec Jeffreys. This was an immigration case involving a family in the United Kingdom where the mother wanted to bring the son to the UK. Things haven't really changed that much. And there was serious opposition that this was not the son of the mother. And DNA fingerprinting proved that in fact the son was who he claimed to be. It was actually a Ghanaian family. Which is why I like talking about it so much. All right. So, okay, so this is all supposed to happen. All right, we just don't have to skip that. All right. So if you expand Francis Collins's representation of the genomic revolution, you really go through some serious landmarks, and these were major advancements in the analysis of the structure and function of DNA, which is what genomics is. It's really the analysis of the structure and function of DNA. And I suspect I have a couple of points here. Can we go back? Let's go back. We can go back. What does it do here? Okay, doesn't do anything. All right, so if you follow 1990, 91, and these were major landmarks. 96, the mouse genome was being sequenced. These were really major landmarks. All the way up to 2000, I think I have a couple of ones here to highlight. This is Bill Clinton getting really excited about the idea. White House press call talking about genomic medicine, which was really exciting. 91 and 2001, and then we go to 2003, when the first draft was really published. So a lot was going on, and you can see all of these are Nature publications. And so these were sort of high impact research that was being done, really analyzing the sequence and potentially the function of DNA. And then the HapMap project followed. We got a lot of information. We know, for instance, that there are 3 billion nucleotides in each cell that we have. And that, as trivial, it will fill this many yellow pages, Very interesting. We knew that there are points in the sequence where individuals sitting next to you is different, right? This became known as single nucleotide polymorphisms, but without showing my age, when I was in my PhD, we called them RFLPs if they could be digested by a restriction enzyme. RFLPs are now single. There is a tautology here, by the way. It's a single variation, but we call it polymorphism, which is many. And so probably the appropriate term is single nucleotide variation, which is what people are beginning to call them now. So we got a lot of information, and there was a lot of promise that as a sickle cell person, it used to be quite amusing if you hear top scientists either on BBC or CNN talk about, now that we have found this gene or this mutation that affects this disease, we can do great things. And I'm thinking, well, for sickle, we found in 1949. We still haven't done anything. And so when we find new information about genetics, sometimes we overreach in what we think it will bring us, and it hasn't always done that. But from the genomic work, we believe that we'll be able to discover and map genes, we can identify candidate genes, we can do diagnostics, we can predict who is going to respond to a particular drug, and that is being done in certain settings. We can do a whole bunch of stuff. This is Francis Collins again, using, this is I think in 2003, a Nature article really highlighting his vision of where this was going to take us. There was one big question that I'll come to in a second. Some of the promises was that we'll be able to use traits to understand complex diseases for which we didn't have a very simple answer to. Again, this kind of, in my view, missed one disease, so I kind of put it there. That's, sickle cell disease. We'll be able to understand sickle cell disease as well, and the interaction of the gene and the environment, and this idea of contemporary genomics challenge. How do you extract everything we know to solve big questions? But there is one big question. And the big question is, well, whose genome did we sequence? Whose genome was it? There were rumors earlier on there was Ronald Reagan's DNA. Then there were rumors there was actually James Watson's DNA. He actually believes it was his DNA. He believes it was his DNA. No, he believes it was his DNA.
It's called narcissism, I think.
Thank you. He actually believes that. He doesn't know that there's somebody in the lab who extracted the DNA. I just showed you DNA all look the same. I always think the technician doing the experiment, I think it should be my DNA. But this is an important question. Whose DNA did we sequence? The HapMap project tried to address that a little bit by studying 5 populations across the world